All right, are there any questions from last time? Yeah, this was right towards the end of the last lecture, right? And I just wanted to, talk about these two things a little bit more before moving on to the next topic, which is about energy considerations in biochemical reactions. Right, I don't see any marker here. Let me see if I have a marker in here. Yeah, I just wanted to elaborate on these items here. See, we said that in addition to serving as the building blocks for DNA and RNA, nucleotides can have many other functions, right? And I wanted to show you how that's possible, right? So let me just show you on the, like, see, we saw the four different nucleotide bases, A, G, C, and T, okay? We also talked about what are called phosphoanhydride bonds, okay? Where two inorganic phosphates, they come together with the elimination of a water molecule, okay? That produces a phosphoanhydride bond. Now, you can have something with a nucleotide, then with three phosphates attached to it, okay? And that becomes a nucleotide triphosphate. That will have two high energy bonds between each of the phosphates, right? So the way the body is typically powered is that this triphosphate is broken down into a diphosphate, right? That's a low energy state. So when energy is needed, you break down the triphosphate to get your diphosphate. And when you need to store energy, an extra phosphate is added onto the diphosphate to produce a triphosphate. Okay, and we are going to encounter this in the next chapter too, right? But I just wanted to mention that. So for some unknown reason, nucleotides appear over there. The phosphate groups are attached to a nucleotide in those molecules which serve to store chemical energy. Then nucleotides are also found in what are called coenzymes. Coenzymes are basically carriers of energy, or they're activated carrier molecules. So let's say, at a particular location in the cell, energy is being generated, right? And it needs to be stored. So to store that energy, you will take that diphosphate, add the extra phosphate group, store the energy, right? Then this triphosphate, it can move around in the cell, and then it can be transported to the place where energy is needed, and then it can be broken down to give a diphosphate, right? And release the extra energy, right? And nucleotides are also used as specific signaling molecules in the cell. And the example of that is what is called cyclic AMP. See, I talked about triphosphates, right? ATP, adenosine triphosphate. If you have an A and then three phosphate groups attached, that's an ATP, high energy molecule, right? The next lower energy molecule is called ADP, right, or adenosine diphosphate, where there will be only two phosphate groups attached, right? If you go down even further in energy, then you're going to have a nucleotide with only one phosphate group attached, and that's called a monophosphate. So AMP is adenosine monophosphate, right? And I'll talk about this, again in the next chapter, today itself, right? So so if in a cell, if you have lots of ATP, right, that means the cell is in a high energy state, okay? On the other hand, if most of these molecules are in the form of diphosphates, means it's in a low energy state. If they are in the form of monophosphate, adenosine monophosphate, it means even lower energy state. Okay, so AMP or adenosine monophosphate is an indicator of the low energy status in a cell, right? And cyclic AMP is something that I will talk about a little bit later in the next chapter, but that is something that actually indicates to the cell that the energy level is very low. And so energy production has to start by burning glucose, by oxidizing glucose, all right, and other sugars. So these are the main points that I wanted to clarify. Okay. Yes, sir. Go ahead. Coenzyme and what? I didn't get you. Coenzyme is what? Activated carrier molecules. Another name for that is coenzyme. Combine a molecule to what? I didn't get you. Oh, I'll give you an example of that in the next chapter, okay? I'll give you an example. Acetyl coenzyme A, I'll give you an example. Yeah. So in the next chapter, these things will become clearer, but I just wanted to, make sense at this stage. That's why I'm just getting a little bit ahead and telling you that this is coming. Alright, so in this chapter, what we have seen is that macromolecules, right? Because I said one uh, distinctive property of living organisms is that they are made up of macromolecules, huge molecules, right? But that can be built up from smaller modules, right? So macromolecules such as polysaccharides, proteins, and nucleic acids that are found in cells, they contain a specific sequence of units that are sugars, right? Monosaccharides, amino acids, and nucleotides. And these huge molecules are formed by these smaller units getting linked together by condensation reactions, where a new unit is added to a growing chain by the expulsion of a water molecule, right? And when you have a huge molecule like this, you have lots of different kinds of interactions, right? So non-covalent bonds such as hydrogen bonds. Hydrogen bond again is the kind of bond that you have between, let's say, hydrogen and oxygen. Like if you're looking at water, I said that hydrogen and oxygen have different affinities for electrons. So even in a covalent bond, the hydrogen will become a little bit positively charged, the oxygen will become a little bit negatively charged. And therefore, between these positive and negative charges, you'll have a weak bond that is formed, which is called a hydrogen bond. So when you have a large macromolecule, there will be hydrogen bonds that will be there in some places between maybe hydrogen and oxygen, or nitrogen and hydrogen, and so on. Okay. Then you will also have hydrophobic forces, because parts of that molecule might really like water, right? There might be other parts that might hate water, right? So all of these different interactions together will determine the final three-dimensional shape of that macromolecule, right? So these specific shapes, as well as the covalent bonds, will allow macromolecules to seek out their appropriate partners and undergo the required reactions with them. Right? Just like you are recognized by your face, all right? People can know you just by looking at your face. All right? Even the macromolecules in the cell, they have to recognize each other by their shape. Okay? If there are two molecules that are going to interact, maybe one fits perfectly into the groove in the other. Okay? Other molecules don't fit, so that makes sure that the reaction is going to take place between the right partners. Okay, any questions on this chapter? Otherwise, we are going to move on to the next chapter, which is about energy considerations in biochemical reactions. And I mean, somebody asked me before, how do you know whether a reaction will take place or not? Okay, that is determined by, the free energy change that is associated with that reaction. Okay? The laws of thermodynamics are not going to be violated in living organisms. Right? They are very much obeyed, but it is the entropy of the living organism and its surroundings that together increases. Okay, the living organism might be generating order. So here we examine the factors which determine whether a particular biochemical reaction is feasible or not. And if a reaction is feasible, okay, the question is what additional factors are needed to get the reaction started. Like say for example, burning a piece of paper, okay, that is a feasible reaction because we all know how to do it, okay. But if you just leave the piece of paper over there, it's not going to burn. You have to apply a match to it. Okay, you have to strike a match and then set it on fire, right? And then the reaction takes place. So you need a kick over some energy barrier to get the reaction going, even if the reaction is feasible. So in this chapter, we'll try to answer some of these questions, right, in the context of biological reactions. Now, it is a fact that the universe is always tending to greater disorder. Okay, and I don't need to convince you about this. Then just try not cleaning your room for a few days. You will see that there is disorder, right? However, a unique characteristic of living things is to create and maintain order. Okay, living things obey the basic laws of thermodynamics. The first law of thermodynamics is that energy can neither be created nor destroyed, but it can only be converted from one form to another. Right? And then the second law is about entropy, or the disorder. The entropy of the universe can only increase. And the entropy is a measure of disorder. So when a cell creates and maintains order, the disorder in its environment increases. And for the cell and its environment taken together, the disorder does increase. So the second law of thermodynamics is not violated by living organisms. Now, living cells perform a never-ending stream of reactions, right? Because there is no rest. As long as you're living, there is no rest. Okay, your cells have to work hard all the time. Okay, there has to be stuff transported across your gut. Your heart has to be pumping and all that. The only time you'll get rest is when you're dead. Okay, so that's real rest, all right? So each cell is like a tiny chemical factory. Thousands of reactions are performed per second. Many of these reactions do not normally occur at biological temperatures, at room temperature, right? So how are such reactions made possible? So these reactions are made possible by using catalysts, which are called enzymes, and also by coupling reactions together. Right? Just like piggybacking, if you want to lift up a bucket of water, okay, you put some water in the bucket, right, it's going to fall, okay, it's not going to come up by itself, but you can link it up with something else, right, so that, piggybacking, you can make the water go against gravity, right, and that's the kind of stuff that the cells are doing all the time. Now, if you look at cells, there are two main types of chemical reactions in cells. The first one are the catabolic reactions. That means breaking down, right? Where foodstuffs are broken down into smaller molecules, generating useful energy, right? And then you also have the anabolic reactions or the biosynthetic reactions, where the energy that is harnessed by the catabolism is used for synthesis of other molecules, right? Because remember, I said that many of the reactions inside cells, when you're trying to form proteins from amino acids, you're trying to form polysaccharides from simple sugars, okay? These are energetically unfavorable reactions. They will not take place by themselves because you're trying to generate order. You're trying to make everything look a lot better than it was before, right? So the way that happens is you couple it with some other reaction. So the energy that is harnessed by catabolism is used for the synthesis of these molecules, right? And the catabolic and anabolic reactions together, they constitute the metabolism of the cell. Right? So you learned something today, right? You hear everybody talking about metabolism. Okay, what is that? That's catabolism and anabolism together. Breakdown of foodstuffs and then manufacture of the biosynthetic components. So to understand what makes a reaction feasible, we have to take a look at the concept of free energy, right? So we know about, the second law of thermodynamics that says that the entropy has to increase, okay? So entropy is basically the, measure of the disorder, okay? So with the disordered stuff, you cannot do useful work. On the other hand, you can look at free energy. Free energy is the amount of energy that is available to do useful work. A reaction is energetically favorable if it proceeds with a decrease in free energy. Right? This makes sense. This is consistent with the second law of thermodynamics. Right? The disorder has to increase. That means the free energy, the energy that is available to do useful work, that has to decrease, right? For a reaction to be plausible or energetically favorable. Cells generate biological order by coupling the biological order generating reactions, which are energetically unfavorable. Formation of proteins from amino acids, of carbohydrates from simple sugars, and then from DNA and RNA from the nucleotide units. All these are energetically unfavorable reactions. So these have to be coupled to other heat generating reactions which are energetically favorable. Right? And releasing heat to the environment ensures that, number one, the entropy of the cell and its surroundings increases, and that biological order is created within the cell itself, even as the environment is messed up more. Now let's look at, all right, any questions so far? So let's look at a few examples of biochemical reactions. The first one is photosynthesis. That's the process by which plants trap energy directly from sunlight to produce sugars. Okay, they are the ones that produce all these carbohydrates. And the reverse process is cellular respiration. Just like you and I breathe, even the cells need to breathe. All right? They have to oxidize carbohydrates or sugars to produce useful energy. Right, and produce carbon dioxide and water in the process. Now there are two main types of reactions that we are interested in. They're called oxidation and reduction. I'm sure you've seen that in your high school chemistry class, even if you didn't do a course on organic chemistry. Oxidation reactions involve the addition of oxygen or the removal of hydrogen. Reduction, on the other hand, involves the addition of hydrogen or the removal of oxygen. That's the first definition of oxidation reduction. Then you can talk about it in terms of transfer of electrons and so on. Okay, and that's what we are going to look at on the next slide, because there may be oxidation and reduction reactions which do not involve oxygen and hydrogen, right? And then you have to characterize that in terms of which one is losing electrons and which one is gaining electrons. Now let's take a little bit closer look now at photosynthesis, right? Now photosynthesis occurs in the chloroplasts of plant cells, right? If you remember, the first day I drew a diagram of a typical plant cell and there was an organelle inside that's called the chloroplast. That's where photosynthesis takes place. The overall reaction for photosynthesis is the following. You have carbon dioxide and water under the effect of sunlight. So you have CH2O from here producing the carbohydrate or the sugar, and then oxygen is released, and then heat energy is also released. Okay? So, I mean, that's why people say, sometimes that plants during the day, they basically give out oxygen, right? I mean, that's why you need plants, right? So, photosynthesis is a two-stage process. In the first stage, energy from sunlight is captured and is transiently stored as chemical bond energy in specialized small molecules. And we'll encounter some of these molecules towards the end of this chapter, right? In the second stage, the energy carriers from the first stage are used to drive a process in which the sugars, like carbohydrates, are manufactured from carbon dioxide and water. And the sugars that a plant produces by photosynthesis are used as a source of chemical bond energy and also as a source of materials to make the many other organic molecules that the cell needs. Because see, if you're having energy trapped, energy from sunlight getting trapped to produce carbohydrates, so you can probably burn this or oxidize that carbohydrate to get carbon dioxide and water and release energy. All right, that's what we do all the time. Okay. So the reverse process of photosynthesis is cellular respiration, and that takes place both in plants as well as in animals. Cells obtain energy by the oxidation of organic molecules, and a very common example is carbohydrates, right? So for carbohydrates, the general formula is CH2O, right? Plus oxygen will give you carbon dioxide plus water plus energy, right? That's the reason why we breathe in oxygen and we breathe out carbon dioxide and water, right? So this is referred to as cellular respiration because oxygen is used up and carbon dioxide and water vapor are released. Also, a great deal of energy is released. If you just go and oxidize sugars like this, you're going to release a lot of energy in one shot. Right? The problem is that the cell would not be able to store all of this energy if this thing occurred in a single step. Right? Therefore, the reaction is carried out in several incremental steps. In fact, if you're looking at glucose, which is C6H12O6, all right, you're going to oxidize that to carbon dioxide and water. There are two separate processes. One is called glycolysis, where you split up the glucose, which is a 6-carbon sugar, into two 3-carbon sugars, all right? That's glycolysis. That's without oxygen. That itself has got 10 different steps in it, all right? I'm not going through the steps. There are 10 different steps, each of them catalyzed by some enzyme, all right? So a little bit of energy is released. That's stored in that molecule. Remember I said about the triphosphate, it's going to get stored in the triphosphate. So the reaction is going to take place slowly so that the energy is progressively stored, right? So each step results in the release of a small amount of energy, and the cell can store this energy in specialized energy carriers. In fact, about 50% of the bond energy that is stored, let's say in the sugar, will be stored in a retrievable form by the cell. And this is a highly efficient process. Okay, 50% of the bond energy is stored. Guess what is the efficiency of the best automobile engine in this world? In terms of converting the energy that is there in the hydrocarbons into mechanical energy. What is the, huh? 20%. 20%, As compared to that, the cell has got an efficiency of 50%. Okay, so I think we have quite a long way to go to figure out how to do things efficiently. All right, then let's discuss oxidation and reduction further. So consider the reaction where you have carbohydrate or sugar, CH2O, being oxidized to produce carbon dioxide and water. So from the preceding definitions, we said whenever oxygen is added, that's oxidation, right? Or removal of hydrogen is oxidation. So from the preceding definitions, the sugar molecule is clearly getting oxidized. Yes, sir, go ahead.
In the preceding slide,
Sure. Let's go.
you say that this reaction it has like a 50% of efficiency. What happens with the rest of the 50%?
Some of it is heat and all that, right? I mean,
Heat.
yeah. Because the wasteful one is basically, production of heat, right? I mean,
Yeah.
see, in your body also heat is being produced because our temperature is at whatever, 98.6, right? I mean, so some of that is needed to maintain the body temperature also, yeah. Yeah, it would be heat, but heat is not, this is useful energy, right? Because this is energy that later on it can break down those triphosphates and get that energy to keep your heart pumping, right? Right, because you need that, you need the energy for everything, even if you want to move, okay?
But then
That also energy is from the triphosphate, yeah.
if you need the heat also, then the efficiency is like higher. No? I mean, if you also can get something from this heat and you need it.
No, but see, for that you'll have to, it's like, if you're going to convert that heat into into energy. Again, I mean, you're going to increase the entropy.
Not so energy, but you said we are I don't know in Fahrenheit, but in Celsius we are like in 37 degrees or something like that.
Yeah, yeah, yeah.
And maybe we need this heat to keep this temperature.
Yeah, yeah, yeah. But we are looking just at the cellular level, okay? When you say you're talking about a human being and all that, okay? But I'm just talking about individual cell. And again, I mean, whether you need heat or not, that depends on the condition outside. Like today, if you step out, I don't think you need heat. If you produce less heat, you'd be better off. But if it's during the winter time, I mean, have you spent any winters here or no?
Not yet.
Yeah, you'll figure out, you do need some heat. Although here it doesn't get really that cold, but it'll be, below freezing, so. All right, now, so the carbohydrate is getting oxidized to produce carbon dioxide and water. Now, alternative definitions for oxidation reduction would involve the loss of electrons or the gain. So oxidation is the loss of electrons, reduction is the gain of electrons. So in a chemical reaction, we know that the total number of electrons wouldn't change. So whenever one compound is getting oxidized, another one will get reduced. And that's why oxidation and reduction will occur simultaneously in a reaction. And these are called redox reactions, right? Reduction oxidation reactions. Now the terms oxidation and reduction apply because, so now from adding oxygen, removing hydrogen, we've now gone to adding electrons, right? Removing electrons, right? But if you have a covalent bond, it's only sharing of electrons. There's no addition or removal of electrons. So, but the terms oxidation and reduction will apply even when there is only a partial shift of electrons between atoms that are linked by a covalent bond between different elements, right? So such a shift occurs when a carbon atom is covalently bonded to an atom with a strong affinity for electrons, for example, oxygen, right? And the carbon atom gives up more than its equal share of electrons and it forms a polar covalent bond, right? If it's linked with oxygen, it becomes carbon dioxide, so carbon is oxidized. And this is a diagram which shows how methane, the gas methane CH4, is oxidized step by step to give you carbon dioxide. So you have methane here, you add oxygen, that's oxidation, you get methyl alcohol or methanol, right? Again, we have encountered all of this before in the last chapter, so don't say, oh, what is this? Okay, CH3OH, that's methyl alcohol. In the next step, you basically go ahead and remove hydrogen, right? I mean, remove hydrogen and you get formaldehyde. Again, that's oxidation. Again, RC double bonded with OH, that's aldehyde, okay? Then in the next step, you basically add oxygen. Again, oxidation, you get formic acid. And in the next step, you just remove hydrogen, you get carbon dioxide. Okay. So this is an example of a typical oxidation reaction. All right, now we move on to the role of enzymes. Now, chemical reactions will proceed only in a direction which leads to a loss of free energy, right? So we know that from a second law of thermodynamics, the entropy has to increase, the free energy has to decrease. Such reactions are said to be energetically favorable. Even such reactions are not going to occur spontaneously, right? And I gave you the example of setting a piece of paper on fire, okay? It doesn't spontaneously ignite. So the molecules require activation energy, that is a kick over an energy barrier to get the reaction going. In the case of a piece of paper, you need that energy, the heat energy from a match, okay? So one way to try to overcome the energy barrier is to supply heat energy, right? However, living cells have to operate within a narrow temperature range, and such a mechanism may not work, So if, in order to get the reactions in your body going, if we have to heat you up, we'll probably kill you before the reactions happen, right? So there has to be something better than that, right? So the necessary boost over the energy barrier is provided by a specialized class of proteins which are called enzymes. And in the next chapter, we will talk about proteins in detail. They are the most versatile molecules in the cell. Enzymes are very effective catalysts, often speeding up reactions by a factor of as much as 10 to the power of 14. The reactions are taking place in your body every second, okay? That would not take place in thousands of years, okay, if those enzymes were not there. If you lose some of those enzymes, then you're in trouble, okay? That's when you end up in the hospital, right? And then you need different supplements and things like that, right? So each enzyme binds tightly to one or two molecules that are called substrates. And this greatly, and once the enzyme binds, the substrates. This greatly reduces the activation energy of a particular chemical reaction. The reaction can take place. Once the substrates have reacted, the enzyme will dissociate from the products and it is free to bind additional substrate molecules. Because as you know, a catalyst doesn't get used up. Okay, it is going to make the reaction take place. It's freed up and it can be used again. Yes, sir. Go ahead.
So substrate here is enzyme combined with the molecule.
No. It's the molecule that binds the enzyme. It's the thing that the enzyme acts on. Okay. Let's say there is A and B, okay, have to react together. Okay. They are the two substrates. The enzyme is like the middle guy, okay. It will make the two of them react, all right? And the two of them are substrates. They will react, all right?
So molecule is there with substrate.
I didn't understand. No, substrate is something that the enzyme acts on. Okay.
Enzyme and molecule.
No, enzyme is a protein, okay? Enzyme is a protein. It's the good guy that makes the reaction happen. Let's put it this way, okay? A and B want to react, okay, but there's a big barrier, okay? Right? Yeah, or maybe, you know, if it's like two people, they don't know how to talk to each other or whatever. You know, some third guy comes and you know, breaks the ice and gets them going, okay, and then leaves. Okay, that person would be the enzyme then, right?
So substrate here is group molecule.
Substrate would be the things that are going to react, okay? Like two different molecules that are supposed to react, okay? But without the enzyme, they would not react. The enzyme will get the reaction started between A and B, and then the enzyme will leave. Yeah, we'll see some specific examples of this as we go further, okay? Now, as I said, you know, the enzyme is successful in lowering the energy barrier. So if you look at this reaction pathway here, and this is the total energy, right? So this is the energy level of the reactants, okay, this line, this is the energy level of the products, the free energy. So you can see that this reaction is going to be energetically feasible because, you know, the free energy associated with the products is less than the free energy associated with the reactants. So it can go in this direction. However, in order for the reaction to proceed, if you look along this pathway, right, look at the red one, that is in the absence of the enzyme, you will have to go over this energy hill, right? Although this place is lower than that one, you have to cross this hill and then only the reaction will be going, okay? Like in the case of a match, when you're trying to set fire to a piece of paper, right, the match is going to, the heat energy supplied by the match is going to make sure that you cross that hill and then the reaction gets going. Here, using an enzyme is going to lower the energy barrier, okay? The reaction pathway now looks like this, right? So this is not too bad. Earlier on, you would have had to overcome this hill in order to go over there. Now you just have to overcome this smaller hill. So the enzyme has lowered this energy barrier that is needed to start the reaction. Right? Now enzymes are highly selective and each enzyme will catalyze only a particular reaction. So by using different enzymes, a cell can regulate a number of reactions in a somewhat decoupled way. Right? There might be enzyme 1 that is catalyzing reaction number 1, enzyme 2 number 2, and so on. Okay? So by controlling the quantity of these different enzymes, the cell can regulate the production of different molecules. Now enzymes find their substrates through rapid diffusion. In the cell, you know, the things are moving around and they're diffusing, but the enzyme is going to recognize its partner with great specificity. Right? So random collisions between the molecules in a cell result in almost immediate dissociation. Right? If they're not made for each other, they'll not latch onto each other. Right? They'll just, you know, collide and then move on. But if they're made for each other, they'll latch on and then the reaction will take place. So when a substrate, that is the thing on which the enzyme acts, encounters the appropriate enzyme, immediate association takes place. Right? It's like a glove, it's like a hand fitting in a glove. Right? Perfect, made for each other. So several weak bonds are formed linking the two of them together and facilitating the necessary reaction. So, all right, any questions so far? So next we move on to onto a discussion of the feasibility of chemical reactions. Right? See, chemical reactions are energetically favorable if they are accompanied by a decrease in the free energy. That's the second law of thermodynamics. That is, delta G, the change in free energy is negative. Right? Now many reactions inside cells, example the synthesis of complex carbohydrates from sugars, these are energetically unfavorable, okay, because they are order generating reactions, okay, they would never occur by themselves, right? So such reactions will occur only if they are coupled to an energetically favorable reaction such that the combined free energy change is negative, right? And this molecule that I alluded to earlier, right, towards the end of the last chapter, that is adenosine triphosphate, ATP, is a molecule that participates in many coupled reactions. This is like the currency of the cell. Like you do work, okay. I'm teaching here, you're doing some other work, RA work, whatever. But at the end of the day, all that has to translate, at least in this country, into dollars and cents, right? Nobody measures things in terms of hours of work and all that stuff. You know. At the end, it's just currency. If you go to the bank and tell them I worked this many hours and all that stuff, they'll give you nothing. Okay, or you go to the HEB and say I worked very hard doing on my RA work, they'll give you nothing. You need the currency. Okay, and for the cell, across all organisms, it is adenosine triphosphate (ATP). Okay. So ATP is capable of storing a large amount of energy in its phosphoanhydride bonds. So we introduce these phosphoanhydride bonds for a reason. Okay, two inorganic phosphate ions. You remove a water molecule between them. You link the two of them together with a high energy bond that is the phosphoanhydride bond. And the picture of a typical, at least the schematic diagram of a typical ATP molecule is shown here. Right, this is adenosine triphosphate. So this is adenine. All right. Remember that base that we encountered last time, that the nitrogen-containing compound is a 9-member ring, right? It's a purine, right? All right. 9-member ring connected to a 5-carbon sugar, which is ribose in this case. Could be deoxyribose if this was DNA, but this is RNA, let's say. And then at the 3' location, at the 5' location, it's connected to a phosphate. So if you have adenine, sugar, phosphate, that's adenosine monophosphate, AMP. If you have 2 of these phosphate groups attached, it's adenosine diphosphate or ADP. If you have 3 of them attached, that is adenosine triphosphate or ATP. All right. So if you're looking at an ATP molecule, all right, this is a high energy bond, phosphoanhydride bond. This is another high energy bond. All right. So these bonds you can. So if you want to store energy, okay, most of the time it'll be ADP is the low energy state, only with 2 phosphates. You add the 3rd phosphate. Right? You want to store energy, add the 3rd phosphate. You got a molecule of ATP. You can have it freely move around in the cell. When you need the energy, you break this phosphate bond. Okay? Phosphoanhydride bond. So ATP is obtained by attaching 2 inorganic phosphate ions in cascade to the adenosine monophosphate AMP molecule. One or both of the phosphoanhydride bonds can be readily hydrolyzed. Hydrolyze means to break with water. Okay? If you remember, when we were forming the phosphoanhydride bond, that was with the removal of a water molecule. That's why it's called anhydride, right? Taking water out. To break this bond, you just have to add a water molecule, right? And this is energetically favorable because, breaking, whenever you're trying to generate order, that's the thing that doesn't work, right? Spontaneously. But if you want to break this up, you can break it and you can easily get your energy, right? So one or both of the phosphoanhydride bonds can be readily hydrolyzed to yield adenosine diphosphate. That means the one with 2 phosphates here, or adenosine monophosphate, and both the reactions are energetically favorable. Right. Now I want to talk a little bit about, it's not there on the slides here, but I want to talk about adenosine cyclic AMP. See, adenosine monophosphate is AMP. What is cyclic AMP? Cyclic AMP, suppose you've gotten rid of these 2 phosphates, so now you have AMP, right? This is the 5' end, right? Remember, this was being labeled 1', 2', 3', 4', 5'. This is the 5' end. This is the 3' end. If you remember, when you were forming nucleic acids from these nucleotides, what you did was you had a phosphate group attached here, and then the 5' end from the next nucleotide was coming and attaching at the 3' location. I told you that, right? So if it's a cyclic AMP, it's not going to be just adenosine monophosphate. This is A with 1 phosphate. This is AMP or adenosine monophosphate. If it is cyclic AMP, this phosphate here is going to come and latch up onto that 3' location. All right. So that's why it's called cyclic AMP. It's like a cycle. All right. It's not just an adenosine monophosphate. It's cyclic AMP, and that is really a good indicator of the low energy status of the cell. So if your energy level is low, okay, your body is trying to always balance the energy, right? To keep the energy within certain limits, or the level of glucose in your blood has to be kept within limits, okay? Each cell has to have enough energy, okay? There should not be you should not be wasting energy, right? At the same time, you should not be starving for energy, okay? So there has to be a mechanism. So if your cell needs more energy, right, then it could be in a low energy state where some of the AMPs have become cyclic AMPs, okay, and that will indicate to the catabolic machinery of the cell that, hey, start burning up glucose and produce more ATP, okay, you need more energy. All right, any questions? So let me give you an example of a coupled reaction, right, where two reactions are coupled together. There might be a reaction that would not have normally happened, right, because the free energy associated with that reaction is positive, but by coupling it with an energetically favorable reaction, you can make the reaction happen. So consider the condensation reaction where sucrose is formed by the condensation of glucose and fructose, right? Both of these are sugars, right? And the two of them are linked together. Now this is an order generating reaction. So the free energy change associated with this, and this is called with this superscript of 0 is the standard free energy change. I will explain in a few minutes what that is, okay? Because the free energy change depends on the concentration of the reactants. This thing with the superscript of 0 means when the concentration, when the reactants have the same concentration, right? Unit concentration, how much of free energy is being released, okay? So you can see that the important thing to note is that this is positive. That means this reaction will never take place, right? On the other hand, if you hydrolyze ATP, that means you break the last phosphoanhydride bond. That is a very energetically favorable reaction, which has got a large negative free energy change. That's minus 7.3 kilocalories per mole. So you break ATP, you get ADP, the diphosphate, the inorganic phosphate ion is released. Okay. So there is a possibility that you might be able to get this reaction, you might be able to couple this along with this to produce sucrose from glucose and fructose. Right? And that's exactly how the body does that. So the two reactions can be coupled together as follows. First, the ATP is readily hydrolyzed to ADP, right? And the high energy phosphate ion is incorporated into location 1 on the glucose molecule, right? Remember, glucose have 1, 2, 3, 4, 5, 6, okay? So location 1, you put that phosphate on. So you now have a high energy bond, high energy phosphate bond at location 1 in glucose. And the ATP, of course, has been used up. It's now reduced to ADP. The glucose 1-phosphate then can react with fructose, right, to produce sucrose and release the inorganic phosphate ion. Okay. So the free energy change, the total free energy change of these two reactions together is basically the sum of their individual free energy changes, and that's going to be negative because one was 5.5 plus 5.5, the other one was minus 7.3, right? So yeah, go ahead.
PI here is
PI is the inorganic phosphate ion. Ion, yeah. It's derived from phosphoric acid, right? If you look back in the notes, I don't know whether it is from last time or the time before that, right? From H3PO4, we can get an inorganic phosphate ion, right? Yeah, it is there in your notes, okay? So from H3PO4, if you take out 2 of the hydrogens, whatever is left over, that is the inorganic phosphate ion. So since the free energy change for sequential reactions is additive, the standard free energy change for this overall reaction is minus 1.8 kilocalories per mole, and this is negative, right? So that means the combined reaction is energetically favorable. The net result is that sucrose is made in a reaction that is driven by the hydrolysis of ATP. All right, any questions?
if the overall free energy change is the positive, it will not be take place, right?
No. Yeah, it cannot take place. If the overall free energy change is positive, then it cannot take place because otherwise it's going to be violating the second law of thermodynamics. All right, any other questions? So now, as I mentioned a short while ago, I said that I was going to talk about the standard free energy change. That means delta G with the superscript of 0, right? So the free energy change for a reaction depends on the concentration of the reactants, right? How much of the reactants you have, how much of the products are accumulating, and so on. So consider the simple reaction below, where you just have one reactant. A is getting transformed to B, right? So this is the reactant and this is the product. The free energy change for this reaction is given by this formula. Delta G, the free energy change is the standard free energy change delta G, G superscript 0 plus 0.616 natural log of the concentration of B, that is the concentration of the product divided by the concentration of the reactant. So here delta G superscript 0 is the standard free energy change. This B within square brackets is the concentration of the product in moles per liter. A within square brackets is the concentration of the reactant in moles per liter. Now if it so happens that the reactants and the products have the same concentration, right, then B and A are the same, right, within square brackets. That means here you're looking at natural log of 1, which is 0. This term drops out. And the free energy change that you have is the standard free energy change. So the standard free energy change for a chemical reaction is the free energy change that you get when the reactants and the products, they have the same concentration. Okay. And of course, you can see that if you look at this equation, okay, if the concentration of the product goes up, okay, what happens? If this goes up, okay, relative to this, okay, what is going to happen to delta G? What? I didn't get you. See, initially this is negative, right? I mean, for the reaction to move forward. So as this goes up, as the more and more product is being produced, right? This thing is not going to be as negative anymore, and then it will become 0. When it becomes 0, that means the reaction will go equally in the forward direction and equally in the backward direction. That's when equilibrium is going to be reached. Okay. So you can use this to find out the equilibrium concentration, because when you set delta G equal to 0, you can solve for the ratio between the equilibrium concentration of B, of the product, and the reactant. And I'm showing this only for a reaction where A is getting transformed to B, but if you have a number of things acting together also, it works the same way. Right? The rate of reaction is proportional to the product of the concentration of the reactants. Right? So at equilibrium, the concentration of the reactants and products does not change, hence delta G is 0. If A sub E and B sub E denote the equilibrium concentrations of A and B respectively, then the formula that you will have for delta G superscript 0, the standard free energy change is minus 0.616 natural log of B over AE. Okay? So if you want a reaction where in the equilibrium stage, the concentration of B, of the product, will be much higher than that of the reactants, this reaction better have a highly negative free energy change. Right? I mean, all right. And this quantity, K, which is the ratio of the equilibrium concentration of the product divided by the equilibrium concentration of the reactant, is called the equilibrium constant of the reaction. Right? So the larger this is, right, the more the reaction will move in the forward direction. Okay, because the delta G superscript 0 is more negative. So all these things make sense. If you're a chemical engineer, I don't know if any of you are. Okay, if you're a chemical engineer, you probably spend a lot more time with these different ideas. Now cells can speed up reactions in at least 2 ways, right? First, what they can do is, see, remember, if you look at this formula, right? If you look at this formula, you can see that, see this delta G0, right? That's the standard free energy change of that reaction, okay? So, as you, like, or if you look at, sorry, not the standard free energy. If you look at this one, okay, see as the concentration of B builds up, okay, the reaction will start going slower and slower in the forward direction, right? So if you can find a mechanism whereby A is getting transformed to B, but as soon as B is being produced, you siphon it away, okay, then you will drive the reaction more in the forward direction, right? More and more, right? So the cells do that, okay? Because what they can do is they can compartmentalize the product of the reaction, they can store it somewhere else, okay? So the reaction can move forward, it's not going to be impeded by the accumulation of the products. So cells can speed up reactions in at least 2 ways. Number one, compartmentalization, where they rapidly remove the products into a separate compartment. And the second way is to use the product. Instead of removing them, if you make the product immediately react with something else and disappear, that'll also work, right? So use the products of one reaction as the reactants of an immediately following reaction. And in the second case above, cells can cause the energetically unfavorable transition X to Y to occur if an enzyme catalyzing the X to Y reaction is supplemented by a second enzyme which catalyzes the energetically favorable reaction Y to Z, right? So it's a coupled reaction. And if the overall free energy change for the reaction from X to Y to Z is negative, then the reaction X to Y will proceed rapidly, although by itself it would have been thermodynamically impossible. So we are getting a glimpse of how cells get reactions going. Yes, sir. Go ahead. Yeah.
So even XY is a positive energy change.
Yeah, X to Y can be positive energy change, okay, but if Y to Z is negative energy change, and that counteracts this positive energy change, like if this is minus 3, I'm sorry, if this is plus 3 and that is minus 8, let's say, then the overall free energy change is negative and it will go from X to Z. Cells do that all the time. All right, any other questions? Okay, now we will get to your question. You asked me about coenzymes and all that. We will talk about that, okay? So activated carrier molecules are also called coenzymes, right? And they play an important role in biosynthesis. The energy that is released by the oxidation of food molecules is used in the construction of other organic molecules. And the energy is temporarily stored as chemical bond energy in activated carrier molecules, right, or coenzymes. An example of that is ATP, right? ADP gets charged up with the addition of that phosphate group, right, to produce ATP. Then it's an activated carrier molecule. It's a guy with high energy. Okay, it can go somewhere else and release the energy. So these molecules can transfer energy from one side to another by rapid diffusion, right? It's like having a battery. Okay, if you just have the regular power system, it's like all fixed in place. But if you want to move energy from here somewhere else, right, without transmission lines, you get your batteries charged up and just carry the batteries and take it over there. Okay, ATP is similar. So these molecules transfer energy from one side to another by rapid diffusion. The coenzymes will store their energy either in high energy bonds, such as phosphoanhydride bonds in ATP. I gave you an example of that. Or as high energy electrons in another class of molecules that we are going to discuss shortly. Any questions so far? So among activated carrier molecules, ATP is the most widely used activated carrier molecule. You can have GTP. Remember, there are 4 different nucleotides, A, G, C, and T. Okay, there is something called guanosine triphosphate too. Okay, that plays an important role in cells. But ATP is the most widely used one. The hydrolysis of ATP releases energy for doing cellular work and biosynthesis, right? Because you can easily break down ATP into the diphosphate and the inorganic phosphate. And the reverse reaction forming ATP is driven by energy from sunlight, right? Or by the oxidation of glucose. When we oxidize glucose in our bodies, that produces a lot of ATP. And these reactions together constitute the mechanism by which cells are powered. All living organisms on this planet, okay, the cells are powered from the simplest bacterium using this ATP-ADP cycle. Right? It's called the cycle because if you want to store energy, convert ADP to ATP. If you want to use up energy, break it this ATP, and it's happening all the time. If your cells stop doing that, you're dead. Okay, so be in the hospital and you'll be dead pretty soon. So, all right. So next, I'm going to discuss this again next time. I just want to show you the picture, right? But there's quite a bit of detail. I just want to highlight the important point. I'm stopping here for today, but I just want to show you the picture. Okay, this is another class of important activated carrier molecules. I'll go over these slides again next time. See, the difference between this and this is that in this location, okay, you have this. To go to the one on the right, that's the charged up one. You put in 2 high energy electrons, okay, to this, and you get this molecule, right? Forget about all of this other stuff. Everything else is the same. You put in 2 high energy electrons, right? You basically get this form of this molecule. So if this one has these 2 high energy electrons, that means this can supply those high energy electrons, okay, for reactions where electrons are needed. Okay, so this would be an activated carrier molecule, and I will talk about this next time. Again, in this, notice that there is a nucleotide. This has no function, right? But for some reason, in this activated carrier molecule, you have adenine, ribose, and ribose phosphate. Okay, the real function is coming from here. If you add 2 high energy electrons, right, then you get this. Okay, and this is another set of molecules that are. I'm sorry, by not just 2 high energy electrons, by adding a proton, which is a hydrogen ion, and 2 high energy electrons, you obtain the thing on the right. Okay, and let me elaborate on this next time. The rest of it is just terminology and all that, but the important thing is that difference that you put in 2 high energy electrons. So if you have high energy electrons, you can store it in there. You can get the charged up form, right? Later on, when you need some reaction, need high energy electrons, then the guy on the right can supply those high energy electrons, right? So this is one of them is called NADP and the other one is called NADPH. So I will talk about this next time. It will take me a little bit of time, and then we'll go on to the chapter on proteins. All right. So any questions so far?